Hello everyone, welcome to my channel Biochemistry by Dr. Tejas Shah. Today we'll see about the disorders of sulfur containing amino acids. This presentation is in the continuation of my previous video that is on metabolism of sulfur containing amino acids. So let us first recap the quick overview of the metabolism of sulfur containing amino acids. So methionine is converted to the active methionine, that is S-adenosyl methionine, further undergo the transmethylation reactions. Then it gives rise to the homocysteine. Homocysteine further converted to the cystathionin. Cystathionin is broken down to form homoserine and cysteine. Homocysteine again converted back to the methionine with the help of vitamin B12 and tetrahydrofolic acid. So now let us see the list of disorders associated with the sulfur containing amino acids. First is the homocystinuria. Second, that is cystathioninuria or cystathionemia. Then hypermethioninemia. Then defect in the absorption, we have Cystinuria and Oxidase syndrome. Lysosomal storage disease, in which the list is the Cystinosis. And rare is the hereditary sulfite oxidase deficiency. So this is the list of the disorders under the sulfur containing amino acid metabolism. So let us first see about the homocystinurias, that is of the classical variety. It is autosomal recessive disease. Here the deficiency of cystathionin beta synthase. So if we see in these reactions, so homocysteine is converted to the cystathionin by the enzyme number 5, that is cystathionin beta synthase, which requires pyridoxal phosphate as a cofactor. So in this condition, this enzyme is deficient. So ultimately there is increased accumulation of the homocysteine level, and further homocysteine is converted to the methionine. So methionine level is also increased, provided the vitamin B12 and tetrahydrofolic acid is sufficient and the enzyme which convert this reaction is sufficient. So normal level of homocysteine is 5 to 15 micromole per liter. But here in this condition, the level is increased up to 50 to 100 times than the normal level. Now in the non-classical type, there is deficiency of either N5-N10 methylene tetrahydrofolate reductase, deficiency of vitamin B12 or deficiency of folic acid. All can lead to the non-classical homocystinurias. So if we see in this reaction, which is very important for the conversion of homocysteine into the methionine, so the enzyme is N5-N10 methylene tetrahydrofolate reductase, which helps in the formation of N5-methyl tetrahydrofolate, which is very much required for this reaction, which convert the homocysteine into the methionine through the vitamin B12. This reaction we have already discussed under the 1-carbon metabolism, where there is importance of this enzyme. Second reaction here, the slide we have already seen, in which you can see here there is role of the vitamin B12 and the folic acid in the conversion of homocysteine to the methionine. So this deficiency of vitamin B12 or folic acid will lead to also accumulation of the homocysteine. So you can classify the 4 variety of the homocystinurias by this. The type 1 homocystinuria, that is deficiency of the cystathionin beta synthase enzyme. Homocystinuria type 2, that is synthesis of N5-methyl tetrahydrofolate is defective. Deficiency of the methyl B12 considered as a homocystinuria type 3, and defective intestinal absorption of vitamin B12, that is homocystinuria type 4. Now we see the clinical manifestation of homocystinuria. Mental retardation is the important clinical manifestation. Then ectopia lentis, that is subluxation of the lens, is nearly the constant feature in patients over the age of 10 years but rarely seen in adults. So what is the biochemical basis behind it? Ocular zonule fibers are composed of the glycoproteins with a high concentration of cysteine. So decrease in the cysteine level will lead to defective glycoprotein formation, as the deficiency of the cystathionine beta synthase, there is no formation of the cysteine. So decreased cysteine will lead to defective glycoprotein, which are very much required for ocular zonular fiber formation. Put the lens. So this is the biochemical basis behind the ectopia lentis. Next is the skeletal deformity or the osteoporosis. So biochemical basis behind it is excess homocysteine level will interfere with the collagen cross-link formation as well as the fibrillin type 1 formation. So there is ultimately defective extracellular matrix in the bone. So that will lead to the skeletal deformity or osteoporosis. Sometimes it will also lead to the Marfan-like syndrome, that is long limbs or arachnodactyly. Hypopigmentation, the biochemical basis behind it, that is when excess homocysteine level that will interact with the copper of the tyrosinase enzyme, which is important for synthesis of melanin. So there is defective melanin formation, which is important for the pigmentation. So because of this reason, there is hypopigmentation is also seen in homocystinuria. And thrombotic strokes and the coronary artery disease are also the important manifestation where the homocysteine level is increased. So let us understand into the flowchart manner the basis behind the coronary artery disease. So when there is increased homocysteine level, it will accumulate in the extracellular matrix and interfering with the collagen cross-link formation. That will lead to the formation of the homocysteine thiolectone free radical, which thiolates the free amino group in low density lipoprotein particles. So ultimately they are ingested by the macrophage but cannot metabolize. So they create a form cells, which is the basis for the tendency of formation of atheroma and lead to the atherosclerosis. Another reason is the increased homocysteine level will activate the Hugman factor, which increase the platelet adhesiveness, and thus it will lead to intravascular thrombosis. So both these two factors will collectively create a scenario of coronary artery disease. So the high homocysteine level will lead to the coronary artery disease. Now if we see the diagnosis in case of the classical homocystinuria. So there is blood homocysteine and methionine level both are increased while cystine level is decreased because there is lack of cysteine synthesis. So cyanide nitroprusside test, that is the urine test performed on the urine, freshly voided urine. It is a rapid and economical test, a qualitative test to be performed. So cyanide is converting the cystine into the cysteine, and the cysteine which react with the nitroprusside to give a purple hue or the magenta red color. So here the homocysteine level is increased. So homocysteine is converted to the homocysteine and it is excreted in the urine and gives this taste positive. Next is the chromatography, which also useful for the diagnosis of the specific amino acidurias. Fourth is the enzyme analysis of liver biopsy specimen when taken or it is through the culture fibroblast. DNA mutation analysis can also be performed for the enzyme deficiency. Prenatal diagnosis by the enzyme assay of cultured amniotic cells or chorionic villi or by DNA analysis. So these are for the classical homocystinuria. Now for non-classical homocystinuria, blood homocysteine level is increased while the methionine level is decreased because if we see the example of the cobalamin deficiency where the homocysteine is converted back to the methionine with the help of vitamin B12 or folic acid. So if these vitamins are deficient, there is lack of conversion of homocysteine to methionine. So homocysteine level is increased but methionine level is decreased. And at the same time, peripheral blood smear also suggests the signs of the anemia, which is developed by the deficiency of the cobalamin or folic acid deficiency. So if we look at the treatment aspect of the homocystinuria, so there is restriction of the dietary methionine is important because you can see the methionine is ultimately converted to the homocysteine, but homocysteine is not converted further because of the block is here, that is homocysteine beta synthase, cystathionine beta synthase enzyme deficiency. So there is restriction of the methionine is very much important in the diet, but here cysteine is not formed, so cysteine to be provided in the diet. Therapeutic dose of the vitamin B6, which helps to stimulate the residual enzyme activity, and thus the patients are also called vitamin B6 responsive patients. Why therapeutic dose of vitamin B6 here is very important. If you see this reaction, homocysteine conversion to the cystathionine, which requires vitamin B6, that is pyridoxal phosphate as a cofactor, and there are other types that is identified, that where the cystathionine beta synthase residual enzyme activity is still present with having affinity of the vitamin B6. So there the therapeutic dose of vitamin B6, it helps to cure the symptoms. But in vitamin B6 non-responsive patients, the folic acid is added, which can be helpful. Betaine, which contains the trimethylglycine, it also decreases the homocysteine by converting into the methionine. So the rise in the methionine level here by the betaine is raised by the baseline level of the methionine, but it is not cause any adverse effect in the body. And lastly, the ascorbic acid is provided in the diet to improve the endothelial functions, which are hampered by the accumulation of the homocysteine in the extracellular matrix. Second is cystathionuria. It is also called cystathionemia. It is autosomal recessive disease. Deficiency of the cystathionase. Here you can see this reaction. Cystathionine is converted to the homoserine and cysteine by the enzyme cystathionase. But in this condition, this enzyme is deficient, which also requires pyridoxal phosphate. So the symptoms are mental retardation, anemia, thrombocytopenia. Cyanide nitroprusside here is negative because there is no formation of the cysteine or as well as the homocysteine is available. So the dietary restriction of the methionine is very much required in this condition. Therapeutic dose of vitamin B6 here again helps to stimulate some residual enzyme activity, and sometimes it relieves the symptoms. Next disorder is the hypermethioninemia. So here there is deficiency of the hepatic methionine adenosyl transferase. Here you can see the number 1 enzyme, methionine adenosyl transferase, which converts the methionine into the active methionine, that is S-adenosyl methionine, or called ADOMET. So here there is deficiency of this enzyme, so there is increased methionine level. So causes are impaired utilization of the methionine, excessive remethylation of the homocysteine, which will also lead to increased methionine level, and secondary to the hepatic dysfunction, as this reaction primarily takes place in the liver. So it is also the part of the Oxidase syndrome where there is malabsorption of the methionine, that will also lead to hypermethioninemia. So hypermethioninemia usually does not cause any symptoms, but it will lead to the central nervous symptoms or delayed mental formation. Cystinuria, it is also called Cystine-lysinuria. So it is autosomal recessive disorder. It's a part of the Garrot's tetrad that comprise of cystinuria, albinism, pentosuria and alkaptonuria. So here there is no defect in cysteine metabolism per se, but there is defect in the diabasic amino acid transporter. So defective reabsorption of the cysteine, ornithine, lysine and arginine. So symbolized as COLA, C-O-L-A, that is cysteine, ornithine, lysine and arginine, where these four are usually reabsorbed, but there is defective reabsorption, so they are excreted, and that's why collectively it is called Cystine-lysinuria. So cysteine in acidic pH or in acidic medium, it converted into the crystal formation. So that will create the cystine stone in the urinary bladder. Here the cyanide nitroprusside test is positive as cysteine is present. So cyanide converts cysteine into the cysteine, and cysteine is react with the nitroprusside to give the magenta red color or purple hue. So here you can see there is cystine stone in the urinary bladder, and the crystals looks like hexagonal cystine crystals like this. Next is the treatment of the cystinuria. So there is increased water intake to keep the urine dilute. Sodium bicarbonate helpful for the alkalinization of the urine or to create an alkaline medium. So that will increase the solubility of the cystine. And the penicillamine is tried to make the soluble derivative of the cystine if this above treatment failed. Next disorder is the cystinosis, that is cystine storage disease. It is the lysosomal storage disorder. Mutation in the CTNS gene which encodes the novel protein called cystinosin. It functions as a hydrogen ion driven lysosomal cystine transporter which transports the cystine across the lysosomal membrane from lysosomal vesicles to the cytosol. So here there is deposition of the cystine crystals in the reticuloendothelial system organs like liver, kidney, spleen, bone marrow, peripheral leukocytes, lymph nodes and cornea. So if we look into the microscopy, there is cystine crystals seen in the white blood cells, that is leukocytes. In the treatment is same as mentioned under the cystinuria, that is increased water intake and alkalinization medium of by the sodium bicarbonate. And apart from the penicillamine, the cysteamine is also tried, which converts the cystine into the cysteine, which prevents the accumulation. And lastly, the renal transplant, that is if the kidney fails. Last is the hereditary sulfite oxidase deficiency. It causes the sulfocystinuria. If we recall this cystine metabolism, at the end it will form the sulfite, and which upon oxidation it is converted to the sulfate, which help in the formation of the active sulfate in the urine. So here there is defect in this sulfite oxidase enzyme. This enzyme contains molybdenum in its active site, and molybdenum helps in the activity of this enzyme. So this deficiency occurs alone or along with the xanthine oxidase deficiency, because xanthine oxidase also contain molybdenum in its active site or its optimum activity. So symptoms are ectopia lentis, mental retardation and seizures. As the active sulfate is not formed, so there is defect in the glycosaminoglycan formation. So these are the symptoms. And these are the references of my presentation. Thank you all. Like, share and subscribe to my channel. Click the bell button to get all the notifications.